Now as you can see that this was the dominant parent and this one was the recessive one. So when I have got a gamete in the F1 generation, I have got a capital W and a small w. So suppose this is what we have studied till now. Now as we are discussing over here, this is what, now as we are discussing over here that F1 progeny when it is crossed with the recessive parent. So mainly we want to know that what is the recessive, what is the genotype and the phenotype of the F1 progeny. So now if I take the F1 progeny to be the dominant one, that is homozygous dominant, and the parent is the recessive one, this is the parent. So what I will be getting from here, this is capital W, this is small w and small w. So from here I am getting all the four gametes same, means suppose this is black color flower, this is white color flower. So what I will be getting from here, all the flowers are violet. And what is the condition? The condition is homozygous dominant, as I have taken the F1 offspring or the progeny to be the homozygous dominant one. Now if it was a heterozygous one, means suppose I have taken the F1 offspring to be heterozygous and I have crossed it with a recessive parent. So what I will be getting from here, I will be getting from here capital W, small w, small w and small w. So from here I will be getting capital W, small w, capital W, small w. So all the four gametes that I am getting over here, half of them will be violet, this one will be all the lower two gametes will be having small w and small w. So half of them will be violet, this one and this one, and the half of the flowers will be white. Why? Because as you can see, this one and this one combines to form violet, whereas this one and this one remains white. So 50% of the flowers will be white, 50% will be violet. So this is due to heterozygous condition. If the F1 is a heterozygous one, that is this one, and if the F1 offspring is a homozygous one, then it will be capital W and capital W, and from there we will be getting all the flowers to be as violet. So what is the genotypic and the phenotypic ratio? The genotypic ratio, as you can see over here, will be 1:1, and the phenotypic ratio will be 1:1. Over here also the same thing. So this is what test cross is all about. So next one is on the basis of backcross, or what is actually backcross. Backcross, the name as it suggests, when a F1 individual is crossed with either of their parents, means either it can be F1 individual can be crossed with the dominant one or with the recessive one. This is what backcross is. Now apart from backcross, there is also outcross. What is outcross? This is a very important thing. This is not given totally in NCERT. F1 progeny, when it is crossed with dominant parent generation obtained from this cross will possess dominant character. So what, let us take an example. F1 progeny crossed with a dominant parent. All the F1 progeny, suppose I am taking capital W and small w, and I am crossing it with a capital W and capital W. So what I will be getting from here? These are the two gametes, both will be coming, and from here one allele will be coming. So this would be the thing. From here, every time what I will be getting? W, small w. Another time capital W, capital W. So in both the cases, you can see one time it's heterozygous dominant, another time it's homozygous dominant. This is what, and that's why we are saying possesses dominant character only. No recessive character will be produced in the outcross. And over here also the genotypic and the phenotypic ratios will be the same. That is 1:1. Now next thing is the most important thing, that is reciprocal cross. What is reciprocal cross? Sex of the parent is reversed. Sex of the parent is reversed. Characters are controlled by karyogenes. Karyogenes means, I will be coming to that. Are controlled by karyogenes which are not affected. Third point, cytoplasmic inheritance results are affected. After this, helps to evaluate maternal inheritance. So what does this all lines mean actually? Now see, I am giving you a very important example. Suppose over here the parent male is capital T and capital T, and the parent female is small t and small t. So from here what do I get? Capital T, small t. That means all will be tall. Now if I reverse this, so this time the female will be the dominant one and the male will be the recessive one. So what do I get from here? All are tall. So this is what it is being said. Now the things are, characters are controlled by karyogenes. Now if this is a cell and inside this, if this is a nucleus, so inside the nucleus these are genes. So the genes that are present inside the nucleus, those are known as karyogenes, and these are not going to be affected by reciprocal cross. But there are certain genes which are present outside in the cytoplasm. These genes will be having, some of them will be affected, and there